Starting to return. Hello, Raquel. Do you want to start sharing your screen so that we will be ready? And now in presentation mode. So we are restarting. The people is probably coming back from the break. And I start to introduce Raquel Ruevo, which is coming to speak from Genes to Function. Welcome, Raquel, and thank you for being with us and accepting our invitation. The floor is yours.
Thank you. So good afternoon. Well, my name is Raquel, and I work on the CIMAR here in Porto, Center on Research on Environmental and Marine Research. I work with two research groups here on endocrine disruption and emergent contamination and animal genetics and evolution. And today I'm going to present some work we've been developing over these past years on nuclear receptor diversity and function. And I will also take this opportunity to address the opportunities and challenges that nuclear receptors present for toxicity assessment. And we talked about nuclear receptors throughout this meeting, and having a technical problem. But first, I'm going to explain why the choice of the nuclear receptors. Well, these are transcription factors, but they're like a particular kind of transcription factor because they're most of them, not all, are ligand activated. They usually form dimers, either homodimers or heterodimers, and they regulate a wide variety of physiological processes in animals, from growth, behavior, reproduction. And for being ligand activated, they're also highly prone to disruption by environmental contaminants, namely endocrine disrupting chemicals. And this feature makes them highly prone to disruption, but also central actors for toxicity assessment. And this is further enhanced by the fact that there's a really wide variety of nuclear receptors. This is a huge family with a vast number and type of receptors. Some are really well known, and you will probably recognize many of them, like the retinoic acid receptor, the thyroid receptor, the vitamin D, VXR, which is highly known for regulation of xenobiotic metabolism, but also endobiotic metabolism and other lipids and regulation. The PPAR, highly recognized for the modulation of lipids and the reproduction-related estrogen and androgen receptors. So as you can see from this panorama, we have a lot of receptors that regulate many processes in animals. And for this reason, they are already considered in some toxicity assessment strategies. Here I provide some info on the OECD conceptual framework for test guidelines for EDCs. And you can see that at level 2, which is the in vitro assays, they are highly considered for this double-edged reason. They're prone, but they're also really useful to address EDC toxicity. But there's a problem here, is that most testing guidelines are highly skewed toward vertebrates and restricted a number of nuclear receptors. And in these guidelines, vertebrates are highly disregarded. And this is problematic. And this is problematic for two main reasons, which I'll explain next. The first reason is that, as we've mentioned during this meeting, the repertoire, the collection of nuclear receptors is not stable across animals. This is a figure retrieved from the work done by a colleague and former PhD student, where she addressed the catalog of nuclear receptors in chondrichthyes, and she compared with previous knowledge regarding other vertebrates and also the vertebrate tunicates and cephalocordates. And what we observe is that there's a variety, that the repertoire across metazoans, it's not stable. And also, there are some subfamilies that simply do not exist in vertebrates. So if we have invertebrate bias, these will be completely disregarded. Also, even when a specific subfamily is present, its composition is not always stable. Here I have an example with the three well-known receptors for vertebrates: the vitamin D receptor, the liver X receptor, and the farnesoid X receptor that control vitamin D. His name is explicit. The liver X receptor is related with cholesterol, and the FXR with bile metabolisms. And what we observed is that in invertebrate species, we only have one receptor, the ECR, already mentioned by David in his previous presentation, and I will come back to it later on. While in vertebrates, we have like a triad of receptors which have surely different functions. So besides the nuclear receptor composition across metazoans, there's a second issue that also came across recently in one of the talks. It regards the function of the receptors. It's not because the receptor is present in a given lineage that its function will be conserved. So what we do in our lab, we combine phylogenetic analysis with the functional in vitro characterization of nuclear receptors. And for this, we use an established in vitro assay that isolates the ligand binding assay. So these receptors are composed of several regions. The two main regions are the ligand binding assay, which binds to the ligand, and the DNA binding domain, which binds to the specific response elements upstream the target genes. So to facilitate the analysis, we isolate the ligand binding assay, and we use a click methodology with a GAL4 DNA binding domain. Okay, so it's kind of like a Lego strategy. So we just change, we have the system all established. We just change the ligand binding domain we want to test, and we address if upon incubation with specific compounds, we observe the increase in luciferase, which is the target gene expression, is a reported gene expression that would later on induce upon substrate incubation luminescence. So this strategy is quite easy to establish and allows us to easily address several nuclear receptors in parallel. So the first example I'm bringing you today relates with the PPAR gamma exploitation by TBT. TBT is a well-known anti-fouling agent. It's been banned for a long time, but it's still present in the ecosystems. And what we observed is that PPAR gamma, which was previously known to bind to the TBT, I'm sorry, which was previously known to bind to PPAR gamma in humans, was also able to bind to PPAR gamma in amphibians. Yet this effect was not observed in teleost fish. A slight increase in activation could be noticed in non-teleost spotted gar. And curiously, this upregulation effect was retrieved again in chondrichthyes. So we went further and continuing explored other non-vertebrate species. And what we observe is that, in contrast with the tested vertebrates, the sea urchin presented and exhibited an effect, except that it was not as observed for the vertebrates that tested positively reacted to TBT. This same response was observed for the gastropod patella. So what this tells us is that even if these organisms, the presence of PPARs can be retrieved along the wide phylogenetic scale, the molecular function of the receptor is not always conserved and has distinct behaviors across species. It's curious to observe, though, that even though the response observed with a sea urchin seemed opposite to what we observed in the vertebrates that responded to TBT, we still observed subsequent changes in fatty acid synthesis through gene expression analysis, and also changes in the lipid content of gonads, meaning that, of course, this needs to be further explored, but this hints that although the responses from a mechanistic point of view do not seem conserved, some of the end points and the end effects are still linked to lipid homeostasis as it would be expected for a PPAR from vertebrates. Yet here I bring another example of regarding the well-known retinoic acid receptor. And this is a curious example because the retinoic acid receptor for a long time was thought to be absent, to be lost in ecdysozoans. Okay, so it means that it was found and it was acknowledged in vertebrates and Lophotrichozoans. Yeah, in ecdysozoans it was accepted that the receptor was ancestrally lost. However, it was not so recently now, because it's been like 6 years, but it was afterwards found to be present in a basal ecdysozoan, the Priapulid worm, which is this funny looking animal here. And it was not only retrieved, but the RAR from Priapulid did respond to retinoids, albeit with different potencies. You should look at the axis. The potencies are quite different, but it still yielded some kind of response, maybe acting as a sensor instead of a high affinity ligand to the RAR. But it still responded to the retinoids. But on the other hand, the same was not observed when it was tested with organochloride insecticides, which are known to bind and activate retinoic acid receptor in humans. So here again, we denote some differences in the molecular function that should just highlight how we should be aware that the presence of the genes do not always imply similar functions, and these has repercussions for the assessment of toxicity. Finally, I bring just to make my case stronger, I bring another example, which is quite curious because it shows that even within closely related organisms, within lineages, we can have episodes of gene losses and divergent functions. So here you have an example with the PXR, CAR, and VDR families. And what we observe is that the CAR, I'm sorry, it's been a bad week. What we observe is that while the VDR receptor is present in both elasmobranchi and allocephali, elasmobranchi, meaning sharks and rays, do not have the gene for the PXR. This was observed in other teleosts too. But what was curious about this, they were so closely related species, and we went to check the PXR function with allocephali, the chimera, which is an elephant shark. And curiously, what we found is that unlike what we were expecting, the PXR, which is related with toxification mechanisms and is usually highly associated with liver, was highly expressed in skin and gill. And its profile of activation was also different from that of zebrafish. Zebrafish is in blue, the elephant shark is in yellow, and we see differences in the activation profiles and differences in the expression tissues. So this points to distinct functions for this nuclear receptor, probably related to the physiology of the organism. And this should also be considered in toxicology assessment. So what I wanted with this presentation, initial part of my presentation, what I wanted to make clear is that we need to identify and characterize clearly nuclear receptors in order to assess the proper physiology across the animal scale and the vulnerabilities of these nuclear receptors upon exposure to environmental contaminants. But we can also take advantages of these nuclear receptors, and these are the examples I want to show you just in the end, as predictors and prospection tools. And the use of the cellular screening assays can be really useful to predict triggers and to be implemented in parallel with other levels of analysis. So one example relates with the work that David just showed us. So we're using this ECR, this endocrine, ecdysone receptor, which was long described as specific from ecdysomes, and is highly related with molting, development, and reproduction in arthropods. And for this reason, because we thought for a long time it was thought it was insect specific, it was widely used to target of insecticides. So now we're expanding our knowledge, and we decided to use this ECR, which is present in far more animals than previously thought. And we're using this ECR with the gamma ray. So initially, we just did the characterization of the binding affinities regarding the ecdysteroids, which would be the natural hormones that would bind to this receptor, but also to insecticides, because of the reason I just mentioned, because it was heavily used as targets. And next, what we're trying to implement is a screening methodology using this ECR with gamma rays. And here I show the data from our joint PhD student, where we use this ECR system, cellular system, to screen water samples, environmental water samples, to try to detect sites with potential contamination. And this will be cross-checked with other methodologies developed in David's lab. So, but besides this screening of environmental samples, we can also think of backward strategies using these nuclear receptors. That is, to try to use them as tests before introducing novel compounds into the environment for marketing to use in the environment. And here I present, just briefly to finish, an example using antifouling agents that are being developed in the Faculty of Pharmacy here in Porto by a green chemistry group. And the idea is to test these compounds using nuclear receptor assays before they are marketed, to see if they could elicit some kind of activation profile. Of course, this is done in parallel with other tests, and it's not in isolation. And here we chose 3 nuclear receptors for this purpose, and 2 levels, 2 animal models. Of course, ideally, we should expand to more nuclear receptors and organisms, or maybe take advantage of the CQ-PAS to try to streamline this analysis. And just the final example is we could also use this to prospect for drugs for human use. So basically, the idea of my presentation was just to call your attention to the need to improve our basic knowledge on nuclear receptor physiology. We're still lagging, notably in vertebrates. There's a lot of gaps in our knowledge on nuclear receptor and characterization. And here I was just mentioning the ligand binding activation, but there's a whole other world regarding which genes are actually targeted by these nuclear receptors across species, and how do they, the relation, the mechanism of transcription per se, the cofactor recruitments. So we have a really complex responses here that should be addressed, but we have a lot of gaps in this knowledge. And as I said, this is a double-edged knife. So on one hand, yes, nuclear receptors are prone to disruption, but we can always use them as privileged tools to address toxicology aspects and mechanisms. And I just want to thank my colleagues and collaborators, and I'll take your questions. Thank you.
Thank you so much, Raquel. It was really interesting. So I hand it